We chose gummy bears, chalk, and Twizzlers because we can use the colors to show the different alleles and easily move them around to show the steps of meiosis. The biological process of meiosis is important for reproduction and variation in the next generation of offspring. In our problem, we have a pedigree that represents the inheritance of a rare disorder. We have determined the mode of inheritance as autosomal dominant, and in our video, we will show the probability of having 2 children with the disease.
We determined the mode of inheritance as autosomal dominant because it does not skip generations, and it is just as likely to happen between boys as it is between girls. Some examples of autosomal dominant diseases are Huntington's and Marfan syndrome. We will be using gummy bears to represent the alleles. Since it is a dominant trait, just having one allele will cause the disease. A red gummy bear represents the dominant allele, meaning affected, and a white gummy bear represents the recessive allele, meaning a person would have to have 2 white gummy bears to not have the disease. To prove that the genotype of 3-5 is heterozygous and 3-6 is homozygous recessive, we will test the genotype starting from 1-1 and 1-2.
We know that 1-1 is homozygous recessive because the pedigree shows he does not have the disease. 1-2 we know is affected, but we conclude she is heterozygous because some of her children are affected and some are not. Therefore, she would have to have 1 recessive allele to produce not affected children. We know 2-5 is homozygous recessive because the pedigree shows he does not have the disease. 2-4 is the heterozygous because some of their children are affected and some are not. Also, because of her parents' genotypes, the only possible combination of alleles for having the disease is heterozygous. Finally, 3-6 is homozygous recessive because the pedigree shows he does not have the disease. 3-5 is heterozygous for the same reasons as just stated for 1-2 and 2-4.
Now that we know the alleles for 3-5 and 3-6, we can show the steps of meiosis to predict the probability of having 2 children with the disease. We'll start with 3-5. There's 1 red gummy bear and 1 white gummy bear, representing the dominant allele and recessive allele. In interphase, the chromosomes are duplicated. At this point, the cell is diploid. In prophase 1, the mitotic spindle forms. Next is metaphase 1, and the homologous chromosomes line up along the metaphase plate. The homologous chromosomes split in anaphase 1, and finally in telophase 1 and cytokinesis 1, the cell splits, resulting in 2 haploid daughter cells. The same process occurs in meiosis 2, except sister chromatids break apart, and the result is 4 haploid daughter cells, with 2 gametes with a red gummy bear and 2 gametes with a white gummy bear. The same process occurs to form these 4 gametes for individual 3-6. These are all the possibilities of gametes that can be formed from 3-5 and 3-6. When fertilization happens, the gametes will combine and there will be different possibilities for the resulting progeny. We will show these possibilities with a Punnett square. The possible progeny is 1/2 heterozygous and 1/2 homozygous recessive, meaning in one child there's a 1/2 chance of having the disease. To find the probability of having 2 children with the disease, we multiply 1/2 by 1/2 to get a chance of 1/4.